Thank you for the opportunity to speak today. So I'm going to be talking about epigenetics and the epigenome, which has a special place in the omics, because it's at the interface between the genes and the environment. And in fact, it's thought to be a link between the genome, the environment, and disease or the phenotypes we're interested in, and may mediate the long-term impact of environmental exposures on disease. So the epigenetic mark that I'm going to be focusing on is DNA methylation. It's one of the most studied epigenetic mark. It's essentially the covalent binding of a metal group to the cytosine at the CpG or CG dinucleotide. There are 30 million of those in the genomes. 70% of them are methylated. Obviously, their role is on gene expression, since they modulate expression through accessibility of the transcriptional machinery. What's important about DNA methylation is it's a dynamic event. It's tissue or cell specific, and it can be influenced again by both genes and the environment. And the reason we have mostly focused on DNA methylation is because it can be measured reliably, quantitatively, and in the cost-effective manner with these array. Most of the common ones are the Illumina array that mostly we've been using in our population-based cohort. So the field of epigenetic with regards to prediction is very much in its infancy. So we're very much on the discovery phase. And I'm going to show some of the things that we have done. So with regards to integrating DNA methylation data in our population-based cohort, our goal was to discover relationships not only between DNA methylation and disease risk, but also environmental exposures and as well as genetic variation. So we've done a lot of these epigenome-wide association studies, or EWAS. And so we've looked at, again, environmental exposures, and I've noted here a few that I've been associated with, but we're looking at many more. Disease-related risk. I'm going to take blood pressure as kind of the poster child for these EWAS and the lessons we've learned from these. And then we have a paper coming up looking at DNA methylation levels and mapping of cis and trans EQTL. So I just want to contrast EWAS and GWAS because it's very important that has an impact for prediction, and I'm going to try to highlight that impact. So first of all, genetic factors are fixed throughout the lifetime, so there is no assumption of temporality of effects, or even directionality of effects, in fact, and then no issue with the time of sample collection. But when we do EWAS, the collection timing matters, and so we have to think about what is the optimal timing of DNA methylation measures as with regards to the outcome of interest. And then, of course, a lot of issues of reverse causation that need to be carefully assessed. So that's one important point here. Genetic factors can be assumed to be randomly assigned with respect to traits, and so when we have this non-random assignment or population stratification, we can certainly identify it and correct for it. But DNA methylation, on the other hand, confounding is present more often than not. An example, cellular heterogeneity. So, for example, if you look at DNA methylation in the blood, blood is multi-tissue, and so you have, because DNA methylation patterns are tissue and cell specific, you have cellular heterogeneity, and that can confound your associations, and so you have to take that into account, be mindful of it. And of course, confounding is also present through measured and unmeasured environmental factors that also may confound your association with disease. For GWAS, we have patterns of correlation of LD that is well defined in the genetic data. In DNA methylation, we know there is intercorrelation between CpGs, but it's often not well defined and certainly not exploited. And certainly we completely ignore it mostly in our EWAS studies. And also, when we do EWAS studies, DNA methylation is the dependent variable. So if you think about using weights in risk prediction, that has an impact, because here you're looking at DNA methylation being your outcome. So I'm going to show you an example of an EWAS that we've done, and they all pretty much follows the same structure. And I'm going to show you some of the lessons we've learned and how these impact their use for risk prediction. So usually we use a two-stage design with the discovery and the replication, similar to GWAS. Similar to GWAS, we do a lot of work with regards to functional notation. And here, it's very helpful when we interpret EWAS results to have other omics data, and certainly gene expression. To have this triangular relationship is often important to interpret these results of EWAS. What is also important is the causal relationship. As you can see, the association, let me see if I'm going to try. The association here has no direction. And for genetic data, you pretty much assume that gene will influence your blood pressure. But you cannot do the same, especially when you're looking at cross-sectionally. You cannot make the same assumption, because in fact, DNA methylation may influence blood pressure, but you may also have blood pressure influencing DNA methylation levels. So you have to have an idea of the directionality. And so here we use the principles of Mendelian randomization to integrate genetic data as instrumental variable to assign directionality in these relationships. And finally, as I said, to have really a good causal mediation kind of effect, and integrating gene expression, we can also do Mendelian randomization using gene expression as well. So this is pretty typical for a genome-wide association study with DNA methylation. Those are the structure that we usually follow for any trait. So this is what we have for blood pressure. When we actually identified 13 replicated CpGs for systolic and diastolic blood pressure in our EWAS. As you can see, we have a diverse population, and the sample size are fairly sizable. So what have we learned from this? So the first thing we've looked at is whether DNA methylation explains more of the blood pressure variance than genetic loci. Notoriously, genetic variant for blood pressure explains very little of the variance in blood pressure. So the question was, does DNA methylation do better? And so in this case, the answer is yes. DNA methylation score based on these 13 replicated CpG explain about 2% of the variance. When we looked at the genetic risk score for the known blood pressure loci at the time, at the time we had 261, they explain less than 1%, or far less than 1% of the variance. So you could see that DNA methylation, in fact, can explain more of the variance in quantitative trait. And that's true as well and for other traits. That study is done here by my colleague Riccardo Maioni in the LBC 1936. And you can see that for many of the traits, DNA methylation, the variance explained by DNA methylation, which is shown is green, as compared to the genetic risk score, is usually larger, with some exception for LDL, for example, in education. And then when you add both polygenic and DNA methylation scores, then you do also better. The other lesson learned is that many of the identified blood pressure associated CpG are heritable. And in fact, the heritability of these blood pressure associated CpG, sorry. Let's see, try to go back here. The heritability of these associated CpG is around between 30 and 60%. And if you look at epigenome-wide heritability, it's only 12%. We can indeed identify MEQTL for these blood pressure associated CpGs. And in fact, those for which we cannot identify these MEQTL had the lowest heritability. Some of them had actually heritability is in fact 0. So looking now at directionality, you can see that we identified CpGs for which indeed DNA methylation influence blood pressure. That's this forward causality, and we're using instrumental variable using the cis-MEQTL for these CpGs. But we also have a number of CpGs for which we have evidence of reverse causality, meaning blood pressure influenced DNA methylation. And here we use the instrumental variable based on the SNPs that are associated with blood pressure. So not always does DNA methylation influence the trait. I think there is, in this case, quite a few for which we have a reverse relationship. The other lesson learned is that it is often important to have other omics to interpret EWAS findings. So for example, we had this CPG here that was intergenic, for which we had no idea what would be the connection with blood pressure. But once we had superimposing gene expression to that relationship, we could identify this particular gene where the CPG was influencing gene expression, and gene expression of that particular gene was also influencing blood pressure. Note that here, the directionality of these arrows, there is no direction. This is strictly association. So if you want to do directionality, if you ascribe some directionality to these relationship, you have to do something called two-step Mendelian randomization, which we did again for all of our identified CPG, and we could indeed find evidence of causal mediation for this CPG, where you had CPG influencing expression of T-span2, and T-span2 influencing T-span2 expression influencing blood pressure levels. So this is through Mendelian randomization. So can we use DNA methylation for risk prediction? And so the field is very much in its infancy, and I could only find in my field one paper where there is an attempt to do such a thing. So how well does DNA methylation predict cardiometabolic traits? So that particular study was done in the Generation Scotland, where they generated DNA methylation scores for actually 10 cardiometabolic traits, and looked at the predictive value of these DNA methylation score in the separate cohort, the LBC 1936 cohort. And you can see that for some traits, you have a near perfect discriminatory power for smoking, where the AUC is 0.98, while for the other, it's more modest. For obesity, for example, the AUC is 0.67, I'm sorry. And for drinking, it's 0.73, and much less for education, college education, where the AUC is 0.59. If you look at blood pressure, the lipid traits, these are also much more modest, where high HDL, the AUC is 0.7, and so for the others, it's actually less, and certainly for LDL cholesterol, it's much less. So there is some good evidence of predictive power for some traits, but not all of those. So what they then did is they associated this DNA methylation score with mortality, and they looked at they compared these scores to both the phenotype, directly phenotype measure, or the genetic risk score. And you can see that in in two occasions here, for alcohol and education, the epigenetic risk score shows a relationship which is not shown by looking directly at alcohol only, or even or education level. And by and large, epigenetic tend to do better than the genetic risk score, with some exception again in the body fat. So there is an advantage to looking at DNA methylation. One thing that DNA methylation does quite well is predicting age. And so the DNA methylation-based age estimator is our set of CPGs that are selected to best estimate chronological age. So the idea is that most people, indeed, will have their age will be predicted along this axis here, and so. But for some people, their predicted age from DNA methylation will deviate from their chronological age. So here, these people would be epigenetically older, and some people would be epigenetically younger. And then the question is whether this deviation, or which is called age acceleration, with deviation from DNA methylation predicted age from the chronological age, influences a variety of traits and disease-related traits. And in fact, we and others have looked at a number of traits, and we in fact find such association. And the first one was actually reported was accelerated aging is associated with all-cause mortality. So I wouldn't want to give you the idea that there is only one way of estimating age. There is actually many of these epigenetic clocks. The one that's probably the most widely used is the Horvath clock, which is a set of 353 CPGs that was derived from looking at multiple tissue. And again, the prediction here was chronological age from multiple tissue. The other one, slightly earlier than that, was the Hannum clock, which was 71 CPGs that predict chronological age, and in this case, the prediction was done only in blood. And there is newer clocks that not only include in the prediction age, but also a number of other factors, which are clinical factors. So for example, the Levine clock, the prediction is for age plus 10 supposedly clinically important factors for morbidity, for example, glucose, liver enzymes, and so forth. So that prediction is for not only you have age, but also a number of other factors that come into the prediction. There is even a newer one that was just published a couple of months ago, which is GrimAge. Here, the prediction is on chronological age plus mortality or risk for mortality. So you have all these clocks for which you can then correlate, and those correlates with age, and the correlation with age is different for these different clocks. So, for example, the Horvath clock does pretty well for age across the lifespan, while the Hannum does the best in blood. And then you can look at deviation from chronological age, and that's called age acceleration, and relate it to a number of outcome. And you can see that, for example, time to death, the clock that does the best, I believe, where is this? I cannot see it from here. The one that does, here we go. The one that does the best is, in fact, the Levine clock, which makes sense since it was derived somewhat for that in mind to try to find those morbid characteristics that are included in the derivation of these CPGs. So, just to conclude, so genome-wide association for DNA methylation have identified new genomic regions that influence complex traits that are not implicated by looking at only genetic variation. But we have to be very careful in interpreting these associations. DNA methylation score explain a substantial proportion of phenotypic variance and are able to predict health and lifestyle factors with some success. Perhaps there is some occasion where data would suggest a potential application of DNA methylation signatures as proxy of self-reported or unreported phenotypes, such as smoking, for example. DNA methylation age and biomarkers of aging from DNA methylation are perhaps good for identifying anti-aging intervention. That's certainly what Horvath is very much, Steve Horvath has very much been pushing with the use of this clock. But what we have to keep in mind is that DNA methylation is dynamic and it's tissue specific, and the predictive ability of this DNA methylation may depend, and these clocks especially may depend on the characteristics of the population and the tissue in which the score was derived. In fact, if you look at this clock association with different traits, you can see that the same clock may be associated with a trait when that clock is derived from blood, for example, and when it is derived from another tissue, that association may be different. And so, that is to be taken into consideration. And so I'm just going to stop here, and I want to acknowledge the young people who actually do all the work, and Melissa Richard, Tancha Huan, and Simon Lidgard, who actually are leading a lot of projects in the Child Epigenetics Working Group. Thank you.
I think we have time for one or two questions. Yes, Nancy?
So one of the intriguing observations of the age clocks, in particular from methylation, is that you can't create DNA expression clocks that do nearly as well as the methylation.
Right.
And so what additional functions do you think DNA methylation is affecting that make DNA methylation so much better at doing these age and other kinds of predictions?
So that's a tough question. We are trying to use genetic to try to understand what this clock measures. So we're doing GWAS of this clock. We're just starting, so perhaps I'll have a better answer in a few, maybe next year. So we don't really know what this clock are measuring. Certainly DNA methylation and aging, I mean, the aging affects DNA methylation pattern quite tremendously. So perhaps, you know,
It's
that's
both.
maybe the readout.
It's consequence as well
Right.
as cause. Yeah.
Yeah.
So I can maybe add to that, because that all-cause mortality paper, I'm actually the last author on that paper. And so the thing about an age predictor is it's a predictor, it's predicted with error. And so we've now made a better predictor, and now that association with all-cause mortality has gone away, or is non-significant. And so what we're thinking is that, you know, it's what we thought all along, that maybe it's picking up something to do with cell type differences, and as we get a better predictor, that's what's coming in. And so since I've got the microphone, the other point you made was a good one, that the cell type differences, the cell type proportions, when we do an EWAS, we want to get rid of those effects. But in prediction, those effects can be important for prediction. Yeah.
Exactly, yeah.
Okay, thank you very much, Myriam. Okay, our next speaker is going to be Peter Kraft, and Peter's going to be discussing environmental exposures.